All right. Hi, good morning, everyone. Thank you for joining us today in this third webinar in the ACell webinar series, the first in 2023. My name is Josephine Lambong. I'm the Manager of Science and Industry Affairs at the Alliance for Regenerative Medicine. Today's webinar is on the topic of lentiviral vector manufacturing. Over the next hour or so, you'll be hearing about the manufacturing process overview of these vectors, including the upstream production and downstream purification. Its use as a drug substance in CAR-T manufacturing, as well as testing and characterization. The speaker is the lead contributor of Chapter 7 of ACell, who I'll introduce in just a few minutes. For those of you who are just joining us for the first time, or if you're not familiar with ACell, it is a collaborative case study on the implementation of quality by design principles in cell-based therapies. This is a project that ARM undertook several years ago after feedback from our members about some of the CMC issues that the field as a whole is experiencing. It occurred to us at that time that these issues were not unique to just the cell and gene therapy. And in fact, some other segments of the biologics industry had gone through similar growing pains before. So the example of the AMAB document was brought up, and it was really something that was instrumental and one of the things that allowed advances to occur in the monoclonal antibody industry in terms of improving manufacturing quality, optimization, and eventually drive down COGS. So that was the impetus for ARM for undertaking the ACell project. And more recently, last year, ARM published AGene, which is the sister project to ACell, on the best practices document for the manufacturing of an AAV gene therapy. So to make ACell an effective resource and reflective of the ongoing innovation in the sector, we chose to focus on immune cell therapy as a case study. And in particular, we further focused the document on autologous CAR-T cell therapy, given the recent approvals and the number of products that are available to patients in the market. We're very fortunate at ARM to have a large number of subject matter experts from our member companies. There are many contributors to this project from more than 30 ARM members. And I would also like to thank Nimble, who generously provided funding for ACell, which allowed us to employ technical writers, without which we would not be able to complete the project. The document is intended to benefit the entire field, so it is available as open access in our website at this link. This is what the contents of the document look like. As you can see, it covers quite a big scope, starting from regulatory considerations, comparability, standards. Chapters 3 and 4 are more of the foundational principles of QBD. 5 and 6 touches on the starting materials and ancillary materials for cell therapies. Chapter 8, which we covered in a previous webinar, details the manufacturing process for CAR-Ts, and then analytics, control strategy, and last but not least, facility design considerations for cell therapies. Each chapter, to an extent, stands on its own, although there's a common QBD theme running throughout. Today's talk will cover the lentiviral vector manufacturing principles and the examples that are laid out in Chapter 7. The webinar today is the third out of a 7-part webinar series for the ACell educational rollout. There are 6 one-hour webinars scheduled roughly monthly until April this year, covering the various chapters, and it will culminate in a workshop in May or June. The schedule of the webinars will be updated in this link down here. So our hope is that ACell, both the document and the webinar series, can serve as an educational tool to inform on the basics of cell therapy, but it will also inform on the methods used to design and quality. As the industry is growing and many people are coming in rapidly into this field, there are a lot of experienced individuals from pharma that are entering the field who may lack the knowledge of the particular unique challenges of cell therapies. We hope that this document will be used by product developers, educators at university, and we also have the aspirational goal that this will be used to help educate regulators. Before we move on, just one housekeeping item. You can use the Q&A function in Zoom to ask the questions. Please don't submit it through the chat. We'll try to answer as many as we can at the end of the talk, and if we didn't get to your question, we will compile them at the end and post the response in the ACell webinar website. And with that, I would like to introduce our expert speaker for today's webinar. Dr. Kelly Zinn is the head of product quality and product quality lead for the early phase CAR-T programs at Atara Biotherapeutics. Kelly joined Atara in June 2021. Prior to Atara, she worked at Bristol Myers Squibb, previously Juno Therapeutics, as the global product quality lead for early phase CAR-T programs and late stage viral vectors. Kelly attended the University of Minnesota, graduating with a BS in plant biological sciences and a Ph.D. in plant molecular biology. Kelly, thank you again for taking the time out of your busy day to be here today. And I will turn it over to you to start your presentation.
All right, thank you, Josephine. Thank you for the introduction. And I'll start by expressing my gratitude to ARM and Nimble for the opportunity to share today's topics on lentiviral vector as it pertains to CAR-T ex vivo use. And I'd also like to acknowledge the co-authors of Chapter 7, whose expertise was invaluable to the development of the document. And those people include Jeffrey Hung, Pratima Cherukuri, Balwant Patel, James Brown, Sebastian Almeida, and Lindsay Gash. So lentiviral vector drug substance is a critical component of chimeric antigen receptor drug products. And the recently published ACell document honed in its scope from within the wide topics of cell therapy, where live cells are delivered to a patient, narrowed to autologous cell therapy, where starting material is derived from the patient in order to prevent donor rejection and prevent graft versus host disease, further honing into immune cell therapy, where cellular material is composed of immune cells like T cells or natural killer cells to recognize and eliminate diseases, and finally narrowing in on CAR-T cells. And CAR-T cells are T cells that are modified to express a receptor on their surface that recognizes antigens on the surface of malignant cells. And once the receptor binds to a tumor antigen, the T cell is stimulated to attack the malignant cells. So the schematic you see here shows the general structure of a chimeric antigen receptor that contains an extracellular antigen binding domain, a transmembrane domain, an intracellular co-stimulatory domain, and a signaling domain. In CAR-T manufacturing, the CAR-T genes are delivered to the T cell through viral vectors. And this can be either gamma retroviral vector or more commonly lentiviral vector. And so today I'll be discussing lentiviral vector manufacturing, testing, and stability. So lentiviral vectors are so commonly used, in fact, that they've been used in several approved CAR-T products, including Kymriah, Breyanzi, and Abecma. And what's more, lentiviral vectors are also used in at least 50 ongoing or planned CAR-T clinical trials. So lentiviral vectors are based on HIV-1, and they're uniquely suitable for CAR-T transduction because they're capable of transducing both dividing and non-dividing cells. I won't go much into the biology of lentiviruses, but I will briefly talk about the genetic elements used to generate replication incompetent lentiviral vector. So the third generation lentiviral vector is based on transient transfection in HEK293, that's human embryonic kidney cell lines, and physical separation of the vector elements across 4 separate plasmids, and removal of key HIV elements contribute to preventing generation of replication competent lentiviral vector. 4 plasmids are required to manufacture lentiviral vector, and these are the transfer plasmid that contains the transgene. In this case, it would be the CAR-T gene. This expression cassette is flanked by HIV-1 long terminal repeats, and these long terminal repeats facilitate packaging the viral RNA genome in 2 viral particles. LTRs also facilitate reverse transcription and facilitate integration of the transgene into the recipient cell genome. The nth plasmid contains viral envelopes, which are typically derived from the vesicular stomatitis virus, VSVG, and they confer enhanced stability and transduction. The rev plasmid contains HIV accessory proteins, notably the rev regulatory protein, which is required for post-transcriptional transport of viral RNA. And lastly, the GAG-pol plasmid contains structural proteins. These are group antigens referred to as GAG and reverse transcriptase. The upstream manufacturing steps for lentiviral vector include culture of HEK293 cells, plasmid transfection, expansion of vector-producing cells, media exchange, and harvest. And of course, optimizations can be made at each step to customize production and improve yield and improve quality of a product-specific vector. Different subtypes of HEK293 cells can be used. HEK293 is an immortalized cell line that's derived from human embryonic kidneys that's been transfected with an E1A adenovirus gene, and therefore it can produce very high levels of protein. HEK293T is a daughter cell line derived from HEK293 original cell line, and HEK293T was transfected with SV40 large T antigen, and that enables amplification of vectors containing an SV40 promoter, and that considerably increases protein expression levels during transient transfection. So HEK293T is most often used for lentiviral vector production. So there are trade-offs among the common options for upstream processing. For instance, adherent cell cultures facilitate higher lentiviral vector titers, but suspension cell cultures are amenable to larger scale production. Serum optimizes cell growth and enhances vector stability, but serum-free production improves product safety by minimizing the risk of animal-related diseases. And lastly, transient transfections are fast and dependable, but they require a costly plasmid DNA supply, whereas stable lentiviral vector systems are still being developed to overcome inherent toxicity of the VSVG gene. The downstream manufacturing steps for a lentiviral vector typically include clarification, purification, formulation, and sterile filtration. And the goals of downstream purification are several-fold: to minimize impurities, concentrate the vector, and formulate the vector to enhance stability and compatibility with the T-cell transduction step. Now, ultimately, downstream processing still strives to maximize vector recovery, and the challenges of downstream purification include the sensitivity of the lentiviral vector to temperature, pH, and shear stress. Now, downstream processing yields are usually 10 to 25% of the harvest, so it's really important to employ strategies to prevent vector loss. We've discussed some of the optimizations commonly available for upstream processing. Similarly, downstream purification steps can be customized to meet a sponsor's specific needs. Again, the goals of harvest clarification, it's to remove cell fragments and large particles. And in the case of where a suspension cell process is employed, clarification even needs to remove whole cells. So clarification can be accomplished in a variety of ways, including low-speed centrifugation, membrane filters, depth filters, and tangential flow filtration, TFF. Nucleases may be added during the clarification stage or added during early purification stage to reduce residual DNA that is contributed from either the host cell genomes or the plasmids. The goal of the purification step in downstream processing is to remove even more proteins, nucleic acids, and low molecular weight impurities. So common techniques for lentiviral vector purification include chromatography, which can separate viral particles from contaminants using properties such as size, charge, hydrophobicity, and binding affinity. TFF can also facilitate both volume reduction, buffer exchange, and TFF can aid in removal of low molecular weight impurities. The final polishing formulation can be performed by chromatography or TFF. The process may include sterile filtration step either before or after the final formulation concentration processing, and the order of which sterile filtration is performed might improve recovery of the lentiviral vector. And finally, common formulation buffers for lentiviral vector include HEAPs, his hydrochloric acid, both resistant to pH drift with temperature changes, tris or phosphate buffered saline. Recombinant human serum albumin may be added to the final formulation to promote stability, and also sugars can act as cryopreservatives and osmolarity regulators. So as we're talking about manufacturing and formulation, another consideration for the overall development and characterization of a lentiviral vector drug substance is extractables and leachables. Extractables are extracted from the container closure system in the presence of solvent, and they're tested for in the container material. Extractables represent a potential impact to the drug substance, and these extractables are typically considered the worst case. Whereas leachables are compounds that leach into the final formulation from the container as a result of direct contact between the container and the formulation. So leachables are tested for in the final formulation itself, and leachables represent an actual impact to the drug substance. Leachable compounds, they might be elastomeric, polymer, other plastic components, or coating materials, depending on the container or stopper type that's used to hold the vector. And where for a drug product like the final CAR-T, you might perform an extractable leachable studies on all product contacting consumables. Typically for the lentiviral vector drug substance, the focus is leachables from that final container and how those container leachables may impact the vector during storage. But the leachables don't only impact the vector, it's also important to evaluate how the vector leachables may impact the CAR-T product, and ultimately if the vector leachables have an impact on the patient. So a risk assessment approach can be taken to evaluate viral vector leachables, their impact to the vector and their impact to the overall CAR-T product by considering the time the vector spends in the container, evaluating the container type. If it's a CZ vial, plastic bottle, or flexible weldable bag, they'll have different inherent leachable risks. Looking at the profile of the potential substances that are identified as leaching from the vector container, and then evaluating how those impact the CAR-T bioprocess. And then finally, determining the impact to the actual CAR-T drug product through the influence of the vector transduction process time and subsequent wash steps. Similar to a CAR-T drug product, process validation should also be performed for lentiviral vector. And process validation is the sum of studies carried out over the clinical development of a drug substance, which contribute to the overall process knowledge. So this begins with the process design phase, and this is information gained in the activities leading up to GMP production. And process design is composed of activities like process development studies, demonstration runs, process lock runs, and tech transfer. Process characterization is protocol and design of experiment-driven studies to evaluate critical process parameters and in-process controls, such as raw material inputs, the impact of temperature, pH, and time of manufacturing and timing of hold steps, the parameters for clarification or sterilization steps, such as the filter material and pore size, evaluation of chromatography approaches to purification, looking at the range of TFF pressure and flow rates, or centrifugation times and speeds. The process characterization would also evaluate the choice and a range of concentrations of the stabilizers and cryopreservatives in the final formulation, and also evaluating parameters related to the container closure system. Another step in process validation is process qualification, and process qualification also needs to be carried out for viral vector via PPQ runs. And process performance qualification runs show that the equipment and the ancillary systems together can perform effectively and perform reproducibly based on executing to approved batch records and specifications. And finally, viral vector also needs to be monitored even after commercial approval of the corresponding CAR-T product. So you've seen a theme in this presentation that the lentiviral vector drug substance development and characterization has many parallels to CAR-T product. And I'll also point out here that even when changes are made to the lentiviral vector, the FDA has recommended comparability studies be carried out in the CAR-T product with a paired arm study where same donor starting material is transduced with both pre and post-change lentiviral vector. And as we wrap up discussing some of the key aspects of lentiviral vector processing and manufacturing, I want to remind us that we're talking about lentiviral vector through the lens of CAR-T development. And so as the drug product, the CAR-T drug product sponsor, you're most likely focused on the CAR-T cell therapy development. And therefore, many sponsors will engage with external partners to obtain lentiviral vector for their ex vivo CAR-T manufacturing. And there's many benefits to working with a CDMO. Partnering externally can help compress timelines and also allows the CAR-T sponsor to leverage external expertise in viral vector manufacturing. But even if you're engaging with a CDMO who has an off-the-shelf vector product or process, there's always going to be product-specific nuances that have to be identified and evaluated. And so some of the key areas to focus and collaborate on to ensure success in your CDMO partnership include oversight of the process tech transfer. And this tech transfer could come from development space that's either process development from the sponsor or even doing process development at the CDMO and transferring activities from process development to the CDMO's GMP facility. The sponsor is responsible for the release and characterization strategy, and many of the lentiviral vector analytics are product specific. And so therefore method development, method transfer, and method qualification, and even method validation are areas of high interface between the sponsor and the CDMO. And then also, given that the CDMOs have the deep knowledge and experience of their own facilities and their own processes, the CDMOs are a significant resource, and they can possibly even be a partner in authoring regulatory filings. So again, process characterization for lentiviral vector drug substances mirrors that for CAR-T drug product. And this is driven by the regulatory classifications of the lentiviral vector for ex vivo purposes. The EMA considers viral vector a starting material, and the FDA considers viral vector a bulk drug substance, an active pharmaceutical ingredient, or a critical component. So the viral vector needs a whole separate drug substance section. This is a 3.2S in a US regulatory filing. And to enable successful product testing and characterization, a quality by design approach should also be employed for lentiviral vector drug substance. And so in early development, the vector quality target product profile and associated critical quality attributes should be identified. The QTPP is a prospective summary of the quality characteristics of a drug substance that ideally will be achieved to meet the desired quality. And a QTPP takes into account safety and efficacy. CQAs are physical, chemical, biological, or microbiological properties or characteristics that should be within an appropriate limit, a range, or a quantitative distribution to ensure the desired product quality. So QTPPs and CQAs are used to drive both process and analytical development. And the initiation of these strategy documents, they drive a whole cascade of activities that ultimately enable generation of the product specification. Assays for lentiviral vector release testing should be qualified or validated depending on the product clinical development stage in order to demonstrate that the assays will serve their designated purpose and ensure that the data is reliable. The justification of specification for lentiviral vector justifies the product specification acceptance criteria based on regulatory safety guidelines and evaluation of representative material. So that representative material is data obtained from lots used in preclinical or clinical studies, data from lots used to demonstrate the manufacturing consistency, that would be engineering runs, and data from stability studies. So in the next couple of slides, I'll give an example of a typical lentiviral vector release specification and talk about some of the concerns, considerations, references, and regulatory guidance that you can use to determine acceptance criteria. Specification attribute categories include identity, safety, general tests, purity, strength, and potency. And these are outlined in ICH Q5B. Identity attributes for lentiviral vector include the presence of the transgene. And the transgene identity attribute should be specific for the vector and be able to distinguish the vector in question from any other product being processed in the same facility. Envelope proteins might also be evaluated to ensure correct change of custody in a multi-product facility. And the Code of Federal Regulations for the Food and Drug Administration give specific rules for identity testing. Safety attributes for lentiviral vector include viral and microbiological contaminants, and these contaminants could be derived from the facility, the raw materials, or even the lentiviral vector itself. USP, ICH Q5A, and the FDA guidance on replication competent retroviruses can help you choose appropriate methods and sample quantities. General attributes of pH, osmolality, and appearance can help confirm proper formulation of the drug substance, and USP methods can be used. We talk about impurities. The lentiviral vector manufacturing process should be designed to reduce impurities as much as possible through the upstream and downstream manufacturing steps. And it's still important to measure impurities in the final vector drug substance, since these impurities may interfere with analytical methods for both the lentiviral vector and the CAR-T products. And that's often the case with high levels of residual plasmid DNA. What's more, the residuals, they can also negatively impact the T-cell manufacturing process, and lentiviral vector residuals might even pose a toxicity risk to the patients if they're carried through to the CAR-T process in high enough levels. So impurities may be derived from the HEK293T host cell system used to generate the lentiviral vector, and these would include residual host cell DNA and host cell proteins. Other impurities to be considered when developing a specification include residual plasmid that's used for transfection and the residual transfection reagents. Non-functional vector that's composed of broken, immature viral particles, particles with insufficient protein, envelope protein, or viral particles that have failed to package. Those could all be considered impurities. Residuals from the culture media, like bovine serum albumin, and residuals from downstream processing, such as nucleases, need to be assessed. And lastly, Visible particulates are also a concern for lentiviral vector because viral particles can aggregate together, or they could aggregate with residual DNA, and this can reduce the titer. And so that has an impact on the availability of vector for CAR-T transduction. And formation of visible particulates in a lentiviral vector drug substance could indicate issues with formulation and stability. Quantitation of viral particles and biological activity are also attributes on a lentiviral vector release specification. Viral particles can be titered in several ways. There's a physical titer, which is expressed as a number of viral particles per ml and reflects the total number of viral particles present. And so that physical titer could be active or inactive particles. Infectious titer measures how much vector is available to transduce a cell, and that's expressed as transducing units per ml. So infectious titer is typically measured post-transduction and quantifying integration of viral DNA into a target cell genome, or post-transduction by measuring expression of the transgene via flow cytometry in a target cell. An infectious titer is always lower than a physical titer, but infectious titer provides a more accurate representation of the transducing ability of the vector. And so to understand the relationship between particle quantity and particle functionality, a physical titer to infectious titer ratio can be calculated and put on a specification. And an emerging expectation from regulatory agencies is to measure lentiviral vector ability to confer a biologically relevant potency to a primary cell. So this could be a T cell potency readout that would be performed on every lot of vector. And this vector potency is especially an important evaluation for autologous CAR-T products to de-risk any CAR-T drug product manufacturing failures that could be due to a deficiency of the lentiviral vector to confer a functional transgene. So I'm going to hop back to a couple safety topics as they relate to lentiviral vector specification. The safety of a lentiviral vector is assessed throughout the vector manufacturing process and at the CAR-T drug product release. The HEK293T master cell bank and working cell bank starting materials are assessed for viral and microbiological agents. And during lentiviral vector manufacturing, microbiological and viral adventitious agents are tested throughout the process, and they're tested at the steps where the contaminant is most likely to be introduced and detected. So therefore, there's routine in-process testing for things like bioburden and endotoxin prior to the sterile filtration steps. And mycoplasma and adventitious viruses are tested at the vector harvest stage. The finished vector is tested for replication-competent retrovirus, sterility, and endotoxin. The ICH guidelines Q5A-R2 on viral safety evaluation of biotechnology products derived from cell lines of human or animal origin is a good reference for developing lentiviral vector safety strategy. And the end of production cells, these are the spent expanded HEK293T working cell banks that were used for upstream production, are also routinely tested for replication competent retrovirus. And the end of production cells may also undergo a one-time evaluation for adventitious viruses. Along the line of special safety considerations that are particular to lentiviral vectors is replication competent viruses. So replication competent lentivirus could develop at any step during the manufacturing of lentiviral vector and during the expansion of ex vivo transduced cells in culture. And this could happen theoretically due to recombination of viral genes carried within the helper and the transfer plasmids. So as I started off in the talk, I talked about the third generation lentiviral vector, which is designed to be replication incompetent by segregating viral elements across 4 plasmids and removing key lentiviral elements. And the engineering of replication incompetent lenti and retroviral vectors has been successful. And this has been summarized in a paper by Cornetta and co-authors in 2018, where they surveyed 282 transduced cell products that had been screened for RCR and listed in the National Gene Vector Biorepository. And after transduction, all 282 transduced cell products were negative for RCR. But still, the potential for pathogenicity of replication competent retroviruses and lentiviral vectors requires comprehensive testing. And the FDA recommends testing for RCL on master cell banks, vector end of production cells, vector supernatants, and the CAR T products. And the FDA also provides recommendations on the sample quantities for each sample type. For cell substrates, the sample quantity recommended is 1% of a bank or 1E8 viable cells, whichever is less. For supernatants, sufficient vector should be tested to ensure a 95% probability of RCL detection if present at a concentration of 1 RCL per dose equivalent. So in other words, the recommendation is that you test the volume of vector that would be administered in 1 CAR T dose, and that's based on the lentiviral vector volume used at T cell transduction. The required testing for RCL at each step of the vector and CAR T manufacturing process, it can require a significant input of time, material, and funds. So it's really important to plan early for the impact of RCL testing on your sample plan and your batch yield calculations. So moving on, stability testing is conducted throughout the life cycle of lentiviral vector development on both the bulks and the finished drug substances. And stability testing serves to provide key information on how the vector is influenced by temperature and influenced by the storage container. So ultimately, the results of stability studies help to establish the appropriate shelf life of the vector and optimal storage conditions. And I touched briefly on the concept of comparability to assess changes in the viral vector drug substance. And similarly, substantial changes to the lentiviral vector manufacturing process should also be evaluated with appropriate stability studies. Lentiviral vector bulk material that is to be stored after manufacture, but prior to formulation and fill, should be put on stability. Major considerations for a lentiviral vector bulk stability plan are using a representative container if a smaller scale aliquot is to be put on stability. Say, for instance, your bulk is held in a half-liter vessel. You could evaluate and justify a 10 ml container of the same composition and the same closure with a fill that has a proportional headspace and surface area to your full scale. And you could bridge that in order to save material in your stability study. Another consideration is making sure to use representative material in preclinical and clinical studies. And lastly, to set up the bulk stability real-time protocol to cover the expected duration of the time between manufacture to the time that the bulk is initiated for fill finish. The stability strategy for a finished lentiviral vector as a drug substance is usually held to a similar standard to that of the drug product. And major considerations for a finished lentiviral vector drug substance are several-fold. One, it's important to use the same container closure system as that's proposed for the actual storage and distribution of the clinical and commercial material. Stability information on at least 3 batches of viral vector drug substance should be provided during phase 1. A target of 6-month stability data at the time of submission should be submitted in cases where the storage periods of that vector are greater than 6 months. And then the real-time finished vector stability studies, they should be set up to cover either the full development period for the clinical trial phase, or to cover the inventory life of the material. So that's if you go through vectors so quickly that you use up a lot in 1 year, you may be able to have a stability study that's set up shorter than the 3 to 5 year example that I have listed here. And then over the clinical development phase of the drug product, additional stability evaluation is required, even for the lentiviral vector as a drug substance. And that includes stressed and accelerated stability studies and container closure integrity stability evaluation. We've discussed the design, development, manufacture, testing, and stability of lentiviral vector drug substance. And I want to finish this talk by going over the actual use of lentiviral vector drug substance in CAR-T transduction. And the FDA's 2020 considerations for the development of CAR-T cell products, the draft guidance for industry, recommends that sponsors develop a lot release test to determine the vector concentration that can be used to normalize the amount of vector used for CAR-T manufacturing. And this normalization is often implemented as a multiplicity of infection, or MOI parameter, on the CAR-T batch record. And that MOI correlates lentiviral vector titer to volume used in transduction. The MOI is calculated from the lentiviral vector titer, so transducing units per ml, multiplied by the transduction volume and divided by the number of target cells. So the volume can be back calculated by knowing the viral transducing units per ml and the target cell number and the MOI. So that might sound straightforward from a batch record perspective, but how does the sponsor figure out the optimal MOI in the first place? And there's several approaches to this fairly new process and regulatory puzzle. And sometimes there can be a shot on goal approach where a set MOI is determined during process development and carried through to tech transfer. But given the lot to lot variability of both the vector and variability of the donor T cells, it's likely that for every batch of vector, a titrated range of lentiviral vector on primary cells in a scale down model needs to be performed to determine an optimal MOI for that lot. This can be assessed during the process development, but this titration studies might also need to extend into clinical phase in order to characterize a sufficient number of vector lots. And it's possible that titration data from multiple lentiviral vector lots can be combined to determine a set MOI as a manufacturing parameter. So now that we're at the end of the presentation, I want to refer you to several Alliance for Regenerative Medicine publications. Of course, the ACell case study is what we've been focusing on today, but I often refer colleagues to Project AGENE. And AGENE is a case study for in vivo gene therapy. It's focused on adeno-associated viral vectors. And although it's AAV focused, the topics in AGENE include quality by design, upstream and downstream processing, process control strategy, and even reference standards. These are widely applicable to gamma and lentiviral vectors for ex vivo purposes, since from a production and regulatory standpoint, there's a lot of overlap between the two modalities. And with that, I'm happy to take some questions.
Excellent. Thank you very much, Kelly, for your talk. That is very informative. I really appreciate your referring to both of our documents that we put a lot of effort to. So yeah, and with that, so we're happy to take audience questions. Again, as a reminder, please submit them through the Q&A function in Zoom. So I'm just going to start with a few that have come in on the testing side, Kelly. So I think you go through a lot of the specifications. There's a table of the example specifications for the lentiviral vector. There's a question here about what is your opinion as to the benefits of reporting options for the ratio of physical titer divided by infectious titer versus infectious titer divided by the physical titer, which is the physical particle per infectious particle and infectious particle per physical particle. That's, try to say that 10 times fast. But yes.
Yeah, well, and I think the physical titer would always be higher than your infectious titer because you're going to have insufficient particles. It's hard to clear those through your downstream processing. I don't think that there's a difference between how you do the ratio. It's just making sure that you understand what it's telling you. That, you know, it would be less than 1. I think that's really, you know, what the intention is. You want it to be closer to 1, but it's typically going to be less than 1. And I think that's probably why the ratio doing infectious titer over, or excuse me, physical titer over infectious titer is probably the better representation of your particle versus your functionality.
Mm-hmm. Okay, great. Thank you, Kelly. I'm going to move on to, let's see, there's questions about, you know, you talked about leachable and extractables earlier. Could you share your experience strategy around qualifications and routine acceptance of either the final containers or any consumable plastic ancillary materials that's used during the manufacturing? So in particular, you know, for long-term testing strategy, if it's acceptable to use the manufacturer's C of A or any of the report, you know, the leachable extractable reports, or do you have to do any additional testing as the sponsor?
I think the current state, and it would be dependent on the feedback that one got from the health authorities, would be to leverage the C of A's for the vector contacting materials, since a lot of those are from like the blood collection industry, and use those to write a risk assessment. So still like the EL program for your vector is going to need to be driven by a risk assessment. Have those C of A's, and then also maybe consider, because you could do a mock run of vector through all the product contacting, have that in your final formulation, and then consider try to do your leachables would be the whole end-to-end process and how it ends up in your container, and then doing the container stability. Where for like a drug product, you'd probably do an extractables for every single component, like cut them up, put the plastics in different solvents and see. But for vector, and again, depending on like how much time is it going to be thawed in its container before you transduce, how long is your transduction time, you know, how many washes before. It might be a lower risk, all the vector product contacting versus your drug product contacting.
Got it. Yeah.
So leverage C of A's. Do a risk assessment.
Yeah, I think risk assessment is the overarching keyword of ACell here. Yeah. Okay, great. Let's see. There's a question about, in your opinion, where are we with the potential use of a stable producer cell line for LV production to replace the need for transient transfection?
I think there's options out there, and would need to see how it works for your particular titer, because, you know, having to source 4 plasmids is really hard, and having to control your transfection step requires additional studies to know what those ratios are between those 4 plasmids. in addition to like whatever transfection reagent you want to use. So having a stable lentiviral vector line would be great. And if you don't need a really high titer, you might be able to use that. So there's options out there. It just depends on if you want like really big scale and really concentrated vector, it might not be the best option yet.
Mhm. Got it. I think there's a couple questions about like stability and like sampling. So maybe I'll group this all together. Let's see, what is the recommendation for time point 0 for stability? And could you share your sampling strategy as to obviously to save on cost and timing for all the testing that's necessary at any time point?
For stability, generally it's following the ICH time points, even for the lentiviral vector drug substance. I think that's the expectation that the agency wants to see, and having that data set, like the frequency of testing early on is important. If you want to try to do any extrapolation for your shelf life, you need that data. So T0, I would say, is at your data manufacturer as close to it as you can. That might get pushed out depending on a number of factors, but typically leveraging the release data, following up with those ICH timelines. And I think the places you can save material is shortening that stability study based on how quickly you go through vector. So if you have a supply and you're going to use it in a year, do you need to do a 3-year study? Or especially at phase 1. So you can probably start with shorter studies with more time points and extend them longer as you get to understand what your clinical use or your commercial use might be like.
Yeah, great. How do you deal with process-derived residuals coming from the lentiviral production process and its impact on the drug product? Do you assume any clearance on the basis of washing and dilutions?
You can model that. Like if you have a residual, like just a clearance model for your CAR-T drug product, you can back calculate that to start honing in on QTPP, those targets for your impurities. And you can also test as characterization if you have specific concerns because you think you might have a lot of like residual plasmid. It's starting to interfere with some of your CAR-T like VCN assays. You could maybe do some tests in your drug product as characterization as you're developing your drug product, and then decide if it's a drug product CQA. I'm more inclined to try to control it in the vector, reduce it in the vector in that process. And then if you still have a concern, monitor it as a characterization in your drug product. And then constantly go to your CQA risk assessment and see how does the input match the output. But I don't think the agency wants sponsors to have to rely on clearance, because you're still potentially putting a lot of vector in your transduction step. And even if it doesn't carry through to the patient, it still could impact like the activation state of your T cells.
Yeah. Okay, got it. Yeah, that makes sense. How do you prove your method of determining MOI when there is lot-to-lot variability in terms of filing an IND and qualifying a method?
I don't think you would need it in an IND. And I tried to explain that. Like you collect a lot of data in development, but you might not be there yet. So you might have to still do a titration in phase 1. And build that along with your life cycle development of the product. So that might be something that gets like slotted into process characterization between phase 1 and phase 2 or pivotal. So I don't think you need it in an IND, the agencies might ask like what your plan and your approach is, but I don't think you need to have it locked in your batch record at that juncture, like especially at filing.
Yeah. Gonna jump away from testing for a little bit. Let's see, could you share your experience with challenges around tech transfer of new technologies to a CMO? Seems like there's a lot of, the number of providers of these kind of processing technologies across the various unit operations are always increasing, and there's little standardization across the competing device platforms. Do you see this as a problem or a challenge?
I think there's lots of opportunities where you could transfer your process, or the CDMO has their own process. If they have their own process, it's good to find out, do they have a scale down model that they've bridged or qualified against their full scale? And can you look at a smaller scale development grade of an external vendor's material and make sure it works in your transduction? Because you could look at a QTPP for vector and say that it has a good titer, the residuals are low, but you still don't know until you've tried to put it into T cells and into your T cell process, because you might have a vector that you've made GMP with a vendor, and you're changing your CAR T processes and it might not work. So it's always that, and that's not the tech transfer question per se, but I think it starts with evaluating a small scale material, and then making sure that you've evaluated the scale up between like a small scale and the GMP, like what parameters stayed the same between like a development space and a GMP space.
Yeah, absolutely. Do you have an opinion on the methods to more directly test titer versus, for example, flow cytometry methods?
I think a lot of people, and myself, would lean towards like ddPCR methods just for ease and throughput, especially for titer that you might be using, having a method that you want on stability, you want on release, you want operators to be able to, in a QC space, execute well and lots. Something, it's an assay, it's higher throughput for like process characterization. So that would be my preference, because I think they give you similar data, and you're still having to compare that to like your particles, that offset between how many particles are there, how many work.
Yeah. That's great. We have 11:59 right now, so I'm going to answer one more question for you, direct one more question for you, Kelly. What is your recommendation for specification setting with limited amount of data, usually less than 10 lots, using statistical tolerance interval, 95% confidence, 95% coverage, or 95% confidence and 99% coverage?
I guess that depends on what phase too. Like if this is getting towards a pivotal, I think the 95% confidence, 95% coverage should be sufficient, because you still have like one more step that it's going to be controlled at, right? It's going to be an input. Recommendation. I would think 95% confidence, 95% coverage should be sufficient. Again, like you engage with your regulator if you had any questions or particular attributes that weren't meeting that. And I don't think you need to be so stringent on your specs at FIH either. Like, of course, like collect that data. You can have like purity attributes on a spec at phase 1 as report result, as a commitment to collect that data. and then hone in on your statistical approach with like representative lots.
Okay, great. So I think we are at time, and as usual, there's a lot more questions that we didn't get to. So I appreciate, Kelly. Thank you so much for your time today and lending your expertise. And I really want to thank the audience as well for engaging in the Q&A. Again, we'll get back to you. We'll post the responses to the rest of the Q&A on our website. And so with that, I'd like to wrap up, and I hope everyone can join us for our next webinar, which will be on ancillary materials. So thanks again, Kelly, and have a great rest of the day, everyone.
Thank you.